Welcome to another FSC Radio podcast episode. Today, me and Santiago will be interviewing Dr. Elise Laetz about the mysteries of the sea. But our spotlight today won't be on the impressive whales or sharks of the sea. No, we will shift our focus on the little underdogs of the water world, the sea-slugs. And in comparison to their land-dwelling cousins, some of these sea slugs have this amazing ability to photosynthesize. They have been a point of focus in Dr. Laetz's research. But before we start discussing these little fellows, we would love to know more about you. How are you today?
Yeah, I'm doing all right. I have to apologize for a slightly froggy voice. I'm getting over a plague that my daughter brought home from daycare. So,
yeah, that's what usually comes with spring. It's so unpredictable.
You can never tell. So please tell us a bit about yourself. So what is it? Were you always intrigued by this sea life? Like, how did you arrive to this photosynthetic sea slugs?
I was definitely always interested in the ocean and always interested in biology. So I grew up in Los Angeles, California, a kid at the beach, and I was always interested in the creatures that we would find and the different stuff that washed up on the beach. So that's been with me forever. Finding sea slugs happened later in life, though. So I think, like many marine biologists, I originally wanted to study whales or sharks or some of these more charismatic animals. But I think a lot of that came from just not knowing very much about all of the other things that live in the sea. And so as I started to do my education, first a bachelor's in San Francisco in ecology, I started to learn about other groups of animals and algae and these other forms that live in the sea. And then I started to get interested in actually how much we don't know about all of those groups.
So you said you started studying in your bachelor in Los Angeles. Later on, because right now you're here in Groningen, so there's a gap in between. So how did the course go in between?
Yeah, so I'm from Los Angeles, and I went to uni to do my bachelor's in San Francisco, California. And then after that, I wanted an adventure to do something international. So I chose a master's program in Germany, in Bonn, in organismal, evolutionary, and paleobiology. So it was a nice broad program. And there I had two fantastic supervisors who both did marine biology research. So even though the program wasn't marine biology specific, I still had a really nice marine biology curriculum. And that really cemented my love for marine biology in particular. So when it was time to do a PhD, I asked one of those supervisors if I could join her in her group. And her focus was on sea slugs. So throughout the courses, I had gotten to know sea slugs a little bit. I knew there were some amazing research questions that were interesting and that I would want to do. So we wrote a grant application, submitted it, got some funding, and I started doing my PhD there. And from then on, it's been sea slugs, and now some other organisms as well, but mainly sea slugs.
So what are sea slugs? So it's like a slug, but then it's in the sea. Can you describe a bit where do you find this? Is it like on the surface of any ocean? Is it also found in the seas? Where are they?
Yeah, so life evolved in the sea. So when we talk about early life and early animal lineages, groups, we talk about them evolving in the ocean because that's where they were. And so the same is true for gastropods, the group that includes sea slugs and sea snails. They originally evolved in the oceans, but then some groups crawled up onto land or crawled up rivers. And that's why we have them on land and in other aquatic systems like lakes and rivers today. Now, some of those sea snails, lost their shells, becoming sea slugs. And the same is true for the ones living in lakes and rivers as well. So we actually have groups with shells and without shells in all of these places. And you can find them throughout the ocean, throughout lakes and rivers, mostly living on the bottom. But some groups have actually moved into the water column and live free floating.
Sounds like a nice life, right?
Just free floating. But you mentioned swimming, so some of them can actually swim?
Yes, some of them can swim quite actively, and some of them actually spend their entire lives swimming. They never live on the bottom.
So they also reproduce while swimming, floating around, and then they never lay. How do they reproduce? Do they lay eggs? I assume.
So how do they reproduce is a tricky question when we talk about gastropods, but a very interesting question. So gastropods are simultaneous hermaphrodites. And what that means is they have boy parts and girl parts at the same time. It depends a little bit on the life stage, how old the animal is. Male parts and male sex cells are pretty cheap to produce, so you can do that at an earlier life stage. So we usually see that the male parts develop first. They become fertile as males first, and then later on in life, they develop the female parts when they get to a certain age or size or health level. And then they meet a partner. They usually fertilize each other, and then they can lay eggs, and the ones that live in the water lay their eggs in the water. Some of them will attach their eggs to other things on the bottom, but some of them have a completely, we would say, pelagic or living in the water column lifestyle.
I'm thinking, I understand that it's necessary to have male and female, but it's usually, at least in the animal kingdom, what you find is that the female usually spends more energy on the on what it's upbringing, right? So what is the reason for these sea-slugs to go from male to female? Why is it? Is it just like purely evolutionary and it just has to be, or why do they change?
Let's say the same. There is more energy that is required to produce eggs, and that's true in the gastropods as well. Once they do sort of develop the ability to produce eggs, they can still use the male parts. So that's why we say simultaneous hermaphrodites. They have both parts. They can use both parts at the same time once they get to that stage in life. But yeah, the energetic investment in producing eggs is much higher. They have to produce yolk, which is this energy-rich stuff in the eggs for the babies to eat. And that is energetically expensive. That said, many species will produce hundreds of eggs, and then there is no parental investment after producing those eggs.
So they just put the eggs and then just let them be. As we sometimes, I think, with the turtles, the same happens, right? That they live in the beach and then they have to run to the sea.
Yep.
Something similar.
Yep.
So I was wondering what was the reason for losing the shell, right? So some sea snails will have the shell to protect them, but it is also a burden to carry around. But at what point do we know even at what point would they say, oh, well, actually let me lose my shell, this will be benefit for me? When did this happen evolution-wise? And later on, what key differences other than shell and non-shell arise from this difference?
Yeah, I think you hit upon something really important there, where this is a structure for protection. We see that in forms that are alive today, species that are alive today that still have this massive shell. It's expensive to produce, it's expensive to carry around, but it's really, really important if you want to avoid getting eaten. So there is a trade-off here, and you're absolutely right. Different groups have sacrificed that protection at different times. So they all started with shells. In evolutionary history, they all had shells. And then different groups lost that shell at different times, and they've adopted other strategies for defense in place of having a shell. And so those are some of the most interesting things that I think we can research about gastropods in general, because they do lose this big defensive structure and they have to do something else instead. And the mechanisms that they have come up with to defend themselves are incredible. So,
please do elaborate.
Yeah, happily. So we have one group of sea slugs. They're a group called cladobranch sea slugs, and they have kind of the typical slug shape that you would expect, but then they have lots of little finger-like appendages on their backs. And these finger-like appendages are extensions of their mantle tissue. That's their main body tissue. And they contain parts of the digestive gland. So this group of sea slugs feeds on cnidarians. These are animals that have stinging cells. If you've ever been stung by a jellyfish, that's a cnidarian. Sea anemones, cnidarians. So these slugs eat the cnidarians. They get stung. It hurts, and you can see the slug react to getting stung. But they have this amazing ability to be able to take up the immature stinging cells from their prey. They don't digest them, somehow, we don't know yet, and they can put those immature stinging cells in at the ends of these little finger-like appendages, and then they can turn them into mature stinging cells and use those stinging cells for their own defense. So if a hungry fish swims along and tries to bite at this sea slug, it can fire off these stinging cells that it stole from its food to defend itself.
This is very freaky.
That's insane.
It's really freaky, but very, very cool. So that's something we're actually working on in my group at the moment, because we don't really know how they do it. We just know that this group has evolved the ability to do it. And so then, of course, we want to know how.
It is something like ingesting a nail and then getting the nail behind you to protect you from someone attacking you.
Yeah. Through your skin.
So I also assume that a big factor here is that you get the baby cells, but then you also need to reproduce them. So there needs, like, through evolution, there has had to be a lot of trial and error between the growth factors that you give them, or are these growth factors usually common across different types of cells to grow them?
Do you mean with these stinging cells?
Yes.
So we don't actually think that it's growth factors at all. There was a study that came out in 2015, I believe, that showed that it has to do with a pH change in the cells, so that the maturation of these cells has to do with an acidification of the compartments surrounding the cell. So the slug doesn't proliferate these cells, it doesn't make new ones. It has to keep eating anemones to keep refreshing its supply, but then it can mature those immature ones by acidifying this compartment, and then it can use them. And once they're used, they're gone. They're done. It has to go get new ones. So in my group, we're looking at how this acidification process might function.
It is also a bit freaky to me that, like, they don't need to only eat for food, but they also need to eat for defense, which costs energy. Though maybe it's me, but the way that I usually think about predators and prey is that the prey needs to eat something, so then it takes energy from someone. But this is not the case here. It's not really taking energy, right? It's looking at these small cells as a mechanism to defend itself, but it's not gaining any energy from it. In the overall, yes, because it needs to defend itself.
Well, and it is still getting energy.
It's still digesting them?
Yeah, its food source has more to it than just stinging cells.
Okay.
So the really interesting thing about the stomachs of these sea slugs is that they separate out the stuff that can be used for defense, and they digest the rest for energy. So the rest of the proteins and cellular material that's not unripe stinging cells gets digested, and that's what fuels them. So that's one of the freaky defense mechanisms. But almost every group of sea slugs has a different freaky defense mechanisms.
A bit more general, are these mechanisms regional? So these slugs evolve in specific regions, which may give rise to localized environments that promote that, or are they found across many different regions?
I'd say it's not geographic. So you can find the slugs that steal stinging cells in all oceans of the world, from the tropics to the poles. You can find them on all continents. They're all over. They're here in the Netherlands. We have some beautiful ones in our lab right now. But yeah, it's not just limited to stinging cells. There are groups of sea slugs that have a sulfuric acid defense, and we know nothing about it. There are ones that can grow, they're called spicules. So these, they're mineralized structures, so hard structures in a sea slug. They can grow these things and put them inside their mantle tissue, kind of their back tissue, so that if a fish comes and bites down on them, there are 1,000 little needles that they get stabbed by. There are sea slugs that eat really toxic stuff, other animals mainly, and they take the toxins, put the toxins in their own bodies, and even sometimes they can chemically transform those toxins into a more potent form that is even more toxic. So every group that has lost the shell has replaced the shell with something else. And it does take energy to do that something else, but the alternative is getting eaten. So there's a really strong selective evolutionary pressure on being able to defend yourself, because if you get eaten, you don't make babies and your species does not continue.
But there was one sea slug who ate the toxin, right? So
Yeah.
this needs to start somewhere. It feels very freaky how, because the mechanism to actually make useful a toxin, as you guys know who are studying this, it's very complex. So I'm wondering, how do all this arise evolutionarily? So was it just by pure chance that randomly the complex was perfectly in place, and then if you eat the toxin, then it's good? Or how was this adaptation to the ingestion of toxin?
Yeah, I think we should specify that a toxin is just a chemical that affects cell function in some way. So we can have really complicated toxins that work on very specific mechanisms, but there can also just be toxins that change simple cell functions. So we should start with that. But then what we're looking at with the sea slugs and their prey is probably an evolutionary arms race. So the original slug and the original prey probably weren't toxic at all, but the prey didn't want to get eaten, and it was getting eaten, so it probably evolved something toxic, and then the slugs had to evolve to be able to not get poisoned because they still needed to eat. So then the prey was getting eaten, and there was an evolutionary selective pressure on that prey to get more toxic, so it did, and then the slugs had to evolve the ability to resist that new toxin. And when we say toxin, we make it sound like it's one thing, but these are cocktails of lots of different chemicals. And so we're probably looking at this arms race where the prey is trying not to get eaten and the slug is trying to continue eating its prey. And then thousands, tens, thousands, millions of years of evolution work on these processes, and we end up with something quite complicated today.
So is this what is called coevolution?
Yeah.
And can you easily trace... because the system in which they live in the oceans are very complex. There are many organisms, so they don't have to adapt only to one prey, but also to multiple preys. So are there like groups of organisms?
Many of these sea slugs are very specific in what they can eat, and that is probably because of this coevolution of toxins and trying not to get poisoned. So a lot of them are highly specific feeders, and that's fun for us because that's kind of how we find them when we go out. So if you know what the slug eats and you see that underwater, there's a good chance you'll find that sea slug. But they're very specific, and they're probably very specific because you can't be immune to all toxins, right?
Yeah,
these toxins are just chemicals that affect cell function, so you can't be immune to everything. So if you have thousands of years becoming immune to this one type of toxin, you can't just switch your prey.
So so they are dependent on the prey. There is always this balance, right, between how many preys and how many predators there are. Mm-hmm. A bit moving on to what we were wondering how we came across you is about the ability of some sea slugs to photosynthesize. So this is very freaky. We always studied in school, right, that the photosynthesis was a plant thing, something that the plants were doing. They just take energy from the sun. How are these animals able to do photosynthesis? What is this? What is even photosynthesis at this point?
So this may have started as a defense mechanism as well, because if you are the exact same color as your food, you're really hard to see. So a lot of sea slugs will also use camouflage, or blending in, to not get eaten. So one hypothesis, which of course we can't go back in time and prove, but one hypothesis for where this ability to do photosynthesis started was just wanting to blend in with your food so you don't get eaten by something else. But, so the way this sort of works in the solar-powered slugs, actually I should go back and say there are two completely different groups, totally unrelated, that can do photosynthesis. And they can both do photosynthesis through completely different ways. If we go back to the slugs I talked about that can steal the stinging cells, some of them eat sea anemones or corals that have algae that live in the sea anemone or coral. Those slugs eat the tissue from the sea anemone, and they digest the anemone's tissue, but they can keep the immature stinging cells and they can keep the algae that sea anemone had. So it's another thing that they can steal and use for themselves. So that's one way. They steal this symbiosis, and then the algae stays alive. It got stuck in a sea slug, but as long as it has light, it can keep doing photosynthesis. It may get excreted at some point, or it may just continue to live in the sea slug. We have examples of both of these options. So that's one way. that slugs have acquired this ability to photosynthesize. And we should say it's not the slug doing photosynthesis, it's that they stole an alga that can do the photosynthesis. The other way is even freakier. And this is the main, I guess, group of slugs that I've been working on. These slugs feed on algae, but they don't eat entire algae cells like we would if we were to eat a piece of lettuce or broccoli or something. Instead, they have this really funky tooth that functions like a straw. So it's really long and thin, and it has like a needle-like point on it. And they pierce the algae cells, and they can suck out the contents. So then all those contents go into their stomachs. Once again, their stomachs are really cool that they can digest all the stuff that's not useful other than as food. But they can actually keep the chloroplasts from those algae cells. They take those chloroplasts, they put them into specialized digestive tissue, but they stop the digestive process, and they can just kind of keep these hijacked chloroplasts functioning in their bodies for a certain amount of time. They can't do it indefinitely. They do have to replenish their chloroplasts. Depending on which species, some can do it for really long periods of time, like up to 10 months. And then other sea slugs can only keep chloroplasts for a couple days. Some can't do it at all. They just digest them immediately.
This is freaky.
Yeah, they're freaky. So they're an animal that has hijacked part of a cell of a different organism from a completely different kingdom of life.
So a bit more general, is this unique to sea slugs? So are there other examples of animals?
There is one other example of an animal.
That we know of, right?
That we know of. There is a flatworm that actually lives on the North Sea coast on mudflats, but not our mudflats, unfortunately. But there is one flatworm that is known to do the same process. It steals chloroplasts from a totally different group of algae, but it steals chloroplasts and it keeps those chloroplasts. There are lots of single-celled organisms that are historically very neglected in research, but can do this. So there are lots of single-celled organisms that steal chloroplasts, sometimes more than once. So they'll steal one organism will steal a chloroplast, and then another organism will steal the chloroplast from that organism. It gets really freaky when you get down to the single-cell world, and that is not researched anywhere near enough for us to have a comprehensive understanding of it.
It feels like I feel like it's because it's more simple. It's also smaller, so then it's much more difficult to understand the complexity of the systems, right? Where you can understand the animal kingdom because there's not so many animals, but when you go to single-cell organisms, there's millions of different types.
And I think they've just been historically neglected. We have the tools now, but we have to have the desire and specialists who are doing it, and we have to decide that that's something that we as a research community want to research more. And there are some really cool groups in Europe and around the world that are starting to really research this. But we don't have a textbook chapter yet that explains what these patterns look like in these single-celled organisms. Certainly not in the same way we do for multicellular organisms like these sea slugs.
Do you know why they're underexplored? Is it because they're not as flashy to sell to maybe the audience? Similarly, I'm guessing like to whales and sharks, they're a bit more flashier than maybe some might assume that sea slugs are. Is it the same with the single-cell organisms? Is that why the research has been stagnant?
Yeah, I wouldn't even necessarily say that it is stagnant. I think it is an up-and-coming field, but so I think it's a combination. It isn't as flashy, or I guess it depends who you ask. Yeah. I think it's fantastic, of course. But yeah, I think there's a combination of it's not as flashy. It is harder to sell to a public. I mean, our research is paid by taxpayers, and to some extent, they do want to know why we care about these things. And beyond just, these are really cool systems and we want to understand them. Sometimes when we apply for grants to do funding, we have to justify to our taxpayers why this research matters. So I think that's part of it. I also think historically we didn't have a lot of the tools that we have. I mean, microscopy is 150 years old now, but the kind of microscopy you need to really see these processes is 10 to 20 years old. We didn't have the genetic tools to do these kinds of things. So how do you tell if this microbe stole a chloroplast? Well, you need DNA to be able to identify that chloroplast. And so then we needed libraries of what kinds of chloroplasts all of these microbes have. So I think this research has been going on, and historically it has been part of the research community. I just don't think we've had the tools or anywhere near enough time to explore the diversity to get that textbook chapter to just sort of understand this.
So maybe going a bit back to the stealing of chloroplasts from the sea slugs, I was wondering, because you said that they need to replenish the chloroplast that they ingest. Is it because, well, they will naturally degrade, but do they not have mechanisms to restore their function and repair them? As, for instance, we as humans have the ability to restore our own cells. Is it because they're different kingdom, different cells, so they didn't develop the mechanisms to restore them?
Yeah, very much so. For a long time, that was absolutely the hypothesis, is that these slugs can keep these chloroplasts, I guess I'll put alive in quotation marks, because they're not really alive, they're not really organisms at all, but they can keep them functional for a while because they're supporting them somehow. So they're replacing proteins that get broken. But then when you start to think about the mechanisms, like what's actually happening in the cells that would be needed to support that, you start to see how impossible that kind of is. So if you want to replace a particular protein in a chloroplast, you need to have the gene, you need to know when to transcribe it, how to transcribe it, you need it translated, you need it delivered to the right place at the right time. So that means getting this new protein through all the membranes into the chloroplast. The mechanism starts to get very, very difficult. A long time ago, this was hypothesized for a long time. People started looking for algae genes in the slugs' genomes before we had slug genomes. But people started looking for these algae genes. And at first, it seemed like there were some algae genes there. But it turns out chloroplasts also have DNA. And if the chloroplast is in the slug, you're going to find some algae DNA in the slug because the chloroplast. So we just didn't have the tools. And as we started to build libraries of the genes that are found inside chloroplasts and the genes that slugs have, it became pretty apparent that the slugs actually don't have any algae genes. So there were some cool studies that looked at egg masses that the slugs laid. That has slug DNA, but an egg has never fed on an alga. So there are no chloroplasts, there are no algae in that egg mass. And if you don't find algae genes in the egg mass, the slug doesn't have it. So when you find it in the adult slug, it's a contamination. Because there is algal DNA there, because they have chloroplasts, and the chloroplasts have DNA. So I would say it was 10, 15 years of really exciting research in this field where they thought there were these genes and they thought they could start to explain the mechanisms. But once we got really nice libraries of the genes that would be involved, it all kind of fell apart. And we don't have algae genes in the slugs. So that doesn't explain it.
So then the slugs just try to tell the chloroplast, okay, you stay alive as much as you want, but I'm not doing anything to replace your malfunctioning. So does it do anything to try to sustain and prolong its lifetime?
Yeah, so there are a couple things that are happening. First of all, when we think about the chloroplasts, I think it's important to think about these chloroplasts in their evolutionary history. The original chloroplast was a single-celled microbe that was swallowed by another microbe. So originally it was capable of independent life. And slowly, throughout history and throughout the evolution of plants and algae in general, some of the functions that this independent alga used to have got transferred over to the host cell's nucleus, and it started to become dependent on that host cell, to the point where chloroplasts don't live by themselves right now.
Similar story
in isolation.
Similar story to what we learned in high school about mitochondria.
Exactly. Same story for both of them. Happened at different times, but it's the same type of process. So these chloroplasts have become somewhat dependent on that host cell. But the amount of dependence that they have depends on what type of chloroplast you're talking about. If we look at modern flowering plants, their chloroplasts are incredibly dependent on the cells that they now reside in and belong in and have to be in. But when we look back at some of the early algal lineages, there's less dependence there. So one of the hypotheses for how these chloroplasts continue to function in slugs for really long periods of time is that they are just a little bit more capable of being independent. They don't break as easily, or they're not as dependent on their host nucleus as a lot of the plants we've studied a lot more. So that's one hypothesis. Last year, we put out a really exciting paper. So me and some of my colleagues, mostly located in the US, where we did a study where we looked at the membrane that surrounds the chloroplast inside the slug. And our goal was to see if that membrane is biochemically active. Is it doing anything? Because up until this point, we didn't really have any indication that the slug is doing something to support photosynthesis
You were watching whether it's a shell or whether there is some activity between.
All of the above.
Okay.
So before, it wasn't even clear actually if this membrane stayed around the chloroplast and was intact. We were able to show it is, but we were also able to show that it is biochemically active. So the slug controls the flow of ions across this membrane. And it does so in a specific way that helps photosynthesis. If we break the ability to transport ions across these membranes, photosynthesis stops working a lot faster. So when we look at all of that, we can say that the slug is doing some things to support photosynthesis and continued chloroplast function inside its stolen chloroplasts. I should now say that was in one species of slug. We have no idea if the whole group does it that way. But it might not be as simple as the chloroplast is independent and it can just keep doing its thing for a while, because it looks like there is some slug input as well.
And you expect similarities between other slugs, or?
Tricky question. So we have at least 4, probably 6 lineages of slugs that have independently evolved the ability to take chloroplasts and keep them for a long time. So more than 2 to 3 weeks.
Independently? They develop this
independently.
Wow.
As in they can all do it, but they do it feeding on different algae, and they're not each other's closest relatives. So they've all independently evolved. We have no idea if they use the same tricks, mechanisms to do it. It's possible, but they don't have to. They could do it in different ways.
That's tricky.
Yeah, it's tricky. But that's why I'm hesitant to say that the finding that from our recent study last year, that we have this biochemically active membrane solves how these slugs keep the chloroplasts alive. No, we have a good indication in one species, but there are 7 other species in these 4 to 6 other lineages that might do it differently.
So because you said that you released this paper last year, I assume that one of the things you're researching now is whether this is sustained, whether that is the case for different slugs.
We're definitely interested in seeing if they're using the same mechanism or if they're different mechanisms, yeah.
And you would do that by comparing the active proteins that are involved in the intermembrane, or you're just looking whether there is intermembrane in between them? Because it feels like the first paper is just looking at is there a membrane bioactive, but it could be bioactive in many different ways. So how are you going to be able to differentiate between different kinds of bioactivity of the different slugs, or you're just looking at whether there's a bioactive membrane and that's it?
I mean, I would say we're doing both. Step 1, is the membrane there?
Okay.
And step 2, is it behaving the same way that we've demonstrated now in one of the slugs? And so we can sort of repeat the experiments that we did last year, or we did over a few years and published last year. We can absolutely repeat that in other species now to try to see if this mechanism is used by all of these different species, or if they have stumbled upon the same result using different mechanisms. If they are using different mechanisms, we need some new experiments to figure out how they're doing it. But I would say that's step 3 in the process, and we're not quite there yet.
Can we please, for the non-biologist, go back and what does it mean for a slug to photosynthesize? Can it sustain itself after it's stolen the chloroplasm? Or does it still have to feed on something?
Yeah. So when the slug has access to food all the time, it's not keeping chloroplasts for 10 months. There's a regular turnover. So it takes them up, puts them in its digestive tissue. They stay there for 2-3 weeks. and then they get digested. We did a nice study a few years ago that showed about a 3-week turnover in one species. We don't know about the rest of them, but it was about a 3-week turnover before all of the chloroplasts it had ingested were eventually broken down and gone. And we did that with kind of a simple sounding experiment, but nobody had known before. We took a slug that can eat multiple different kinds of algae. We fed it one kind of algae only for a long time, and then we changed its food. And then we could do DNA barcoding to see what kind of algae it had in its body at different sampling points after we did this food switch. And what we saw is that after about 3 weeks, there was no more of the first algae in its body. So we know all of those, there was no more DNA in its body. So we know that all of the chloroplasts from that first algae that it was eating were gone in that species. That's all we know, though, so far. So yeah.
And can it solely rely on this photosynthesis, or does it also need other sources of food?
No, well, they solely feed on algae. Some species are very specific to one type of algae. Others can eat all kinds of algae. So the algae itself doesn't really answer how they do this either. There are lots of different kinds of algae that could be incorporated, but not in all slugs. And so that's kind of complicated on its own. These slugs get all of their nutrition from algae, but they can't live happily forever on their stolen chloroplasts. It'd be like saying maybe they get 70% of the energy they need, and at some point you're starving to death. You need to feed again, or you're not going to survive.
So I recently had a course that was in bioenergy and metabolism, and one of the things that we saw was that we as humans cannot get all our energy from lipids, so to say, because with these lipids we create a molecule, pyruvate, but this does not give us all the essential amino acids. So what I'm thinking is, are these sea-slugs not able to get all the energy from chloroplasts because it's just energy that they're receiving and they have essential amino acids? Do we know this, or why is it that they cannot only rely on photosynthesis?
We don't know is the short answer. The longer answer is about all we know is that they do get photosynthetically fixed carbon. So that means when we think about photosynthesis, we have light, carbon dioxide, and water getting converted, well, through the energy of light, into sugars and oxygen. So those sugars contain carbon, and that carbon is made available to the slug. There's not enough carbon over a forever type period that the slug can survive forever. We don't know about amino acids. We don't know about nitrogen dynamics and all of the other things in there. We don't know if there are other nutrients that are limiting. It's certainly all plausible. We don't know.
So this is something to research, but in the future, it's not in the in the future. I was just a bit curious about that.
Me too.
Hopefully, we can get an answer soon.
Can we talk about the color of sea slugs? Because when researching this, I was met with a lot of beautiful pictures of so colorful sea slugs. But when you compare them to something you see in your garden, the land-dwelling slugs, they're grayish, they're bluish, they're like a bit brown. So what made them? What has happened in the evolution for them to have this beautiful coloring?
Yeah. So I should say, first and foremost, if you've never Googled sea slug, Google sea slug. Do it. It will be worth your while, and you might spend longer than you anticipated looking through Google Images. I think this all comes back to defense strategies again. The best defense you can have is to never get bitten or never have something try to eat you from the beginning. And one of the main ways that animals do that is communicating through color, through visual cues. One group of sea slugs, actually multiple groups of sea slugs, have taken on different types of coloration as a visual signal. Don't eat me. That's why they can be so brightly colored and so beautiful. They have striking patterns. There have been a lot of studies that look at specific patterns and the avoidance of fish and stuff like that. And they overwhelmingly find that fish can identify these patterns and they know to avoid them. There are even other animals that are totally unrelated, like, for example, a group of sea cucumbers that have gotten these color patterns to look like sea slugs because fish avoid them so much. So they can be a very, very strong visual signal. And of course, that's happening in groups that aren't trying to camouflage with their food. So we just have opposing strategies, and different groups of sea slugs have chosen different strategies to try to not get eaten.
Oh, but then I suppose then do they have more predators than, for example, the land slugs? Why are they so gray? Why are the land slugs so blue, so gray?
Yeah, I think it has to do with sort of the lifestyle, the way these animals live. Coral reefs are brightly colored, for example. So being brightly colored on a coral reef isn't that different. They look like they'd be really easy to find. They're not necessarily very easy to find. But if a fish learns that pattern, and you see that pattern, and the fish quickly can learn avoidance and not feed on those sea slugs. A lot of our snails and slugs on land, I mean, they're also prey. There are lots of animals that eat them. And so that this may come down to having a lifestyle where you hide more so, or the snails certainly rely on their shells for protection. But a lot of the slugs hide. They only come out at night when visual cues don't do anything. I should say our land slugs and snails might be less attractive, but they are also full of toxins. So I think that's quite interesting and something maybe to appreciate them, even though they're not as pretty.
I'm wondering, they take these toxins. So is this one of the mechanisms that sort of nature has found to remove unwanted compounds from soil? This is reminding me. Lately, I've seen some papers in a course that is about catalysis. We may hear about that soon. Regarding PFAS. So PFAS is a compound that has carbon fluoride compound bonds, and these are compounds that you don't really find in nature, and there are no way to find in nature. So always when I was looking at this, the question that came to mind is, well, there are many things that you could think, oh, this compound nature would have not been able to recycle, but there's still ways that it has found. So are these slugs ways that nature have found to recycle byproducts of nature? Or are these toxins usually toxic for some things and not toxic not for others? So they're just taking advantage that some organisms are more toxic to that toxin rather than others. This is a bit of a non-question.
I don't know the exact answer to that. I don't know for land slugs, for example. I don't know if they're taking up toxins from their environment, if they're producing them themselves, or a combination of the two. I think both are absolutely plausible. I think the reason that this probably evolved in the first place is a defense mechanism, because once again, they lost their shells, so they have to do something else. So that would be my guess for what the evolutionary pressure was to get toxic. I don't know if they're taking up chemicals from the environment, though, that aren't needed or wanted or part of other processes.
Maybe this is coming back a bit to the beginnings, but I was just wondering, you keep saying that the sea slugs lost their shells, so then they really need a defense mechanism. But I'm sure some of these shells would have not been so great at protecting the slugs. So some slugs with shell would have also found defense mechanisms through toxins. Or are these toxins only specific to the sea slugs without shells?
No, you're right. It's absolutely, it's not as cut and dry as you have no shell, you are toxic. We have shelled snails that are toxic as well. Toxic metabolites have been found in them anyway. And we also have steps that might be in the middle. We have lots of animals that have shells, but they're very reduced. And so there's not really a protective function, and that might be a character that evolution is kind of weeding out. And maybe in another 10,000 years, that species of snail or its descendants no longer have a shell at all. So we also have these middle stages and of course then those animals are toxic as well as they even though they might still have a small shell or a less functional shell.
So it was probably that when they lost the shell, it was not that from one day they dropped the shell and now they don't have it. It was probably slowly reducing the impact of the shell.
Yep. Should also say that even for sea slugs, when they first hatch out of the eggs, they have shells. So in their larval stages, they have shells. And so when we culture these animals in the lab and when we raise them up from eggs, when they hatch, they look like tiny snails, like you would see on land, but just tiny and transparent. And then one day they all go through metamorphosis, they drop their shells, and all of a sudden they start looking like slugs. And that day is funny because you can look in the dishes that we culture them in and you see tons of these little tiny transparent shells all over the place. Almost looks like sand.
Are these still hard shells even though they're transparent?
Yep. And they probably still provide protection for the larvae. So they're probably still functional in that sort of plankton world from predators also living in the planktonic world.
So coming a bit with this general idea of getting toxins into our body, or even better, photosynthesis. Is it something that is ever discussed that in the future we may find mechanisms to have photosynthesis in our skin, or be able to eat things that are maybe toxins to us, but that we're able to engineer, so to say, this may be too transhumanist, how to reduce those things. Some more biology example I could think of is engineer pathways in our body to create all essential amino acids, and then you can eat only from lipids. But you could also talk about getting chloroplasts in our cells. So is this something that is anyone talking about, anyone thinking of? Or this is so distant in the future that there is no need to talk about it?
So it's funny you mention that because while I was doing my PhD, I was working with another lab, a group in Düsseldorf, Germany, and they had the exact same questions about whether or not photosynthesis could support a human or a cow. And they went on a little adventure to do some math to figure out how much skin a cow would have to fill up with chloroplasts in order to support its metabolic needs. And they calculated it would need 1,600 square meters of surface area, or skin filled with chloroplasts, if it was to produce energy with the same efficiency as corn. So if you can picture a cow with 1,600 square meters of skin, I think you start to see where, metabolic efficiency doesn't work quite the same way in these tiny, tiny animals versus larger animals like us.
So the main point is that the relative surface area to volume ratio is the key factor.
Unfortunately, yeah. So we would need either cows that are really, really wrinkly,
tiny
or
cows.
A lot more efficient than even a plant like corn could be. And certainly I wouldn't want to be a human with all of the extra skin, and I don't think it's going to be very practical or feasible. But the thought experiment is great, and I'm really happy my colleagues sat down and did all of this math, because it brings up some funny mental images.
What about in taking toxins? So, like I know I mentioned before with the lipids, but I can also think of eating plastic. Right, we have a lot of issues with eating microplastics, but if we find a way to digest microplastics, then that issue would be solved. Of course, there would be a lot of issues with it still, but is that something that is also thought of? Or when you're looking at these mechanisms of introducing toxins into the body?
I would say that's not a field that we are actively researching in our group, so I can't really speak to that as an expert. But I do know that there are organisms that can consume plastics. So there are lots of different bacteria, and there's some really nice research groups that are looking at plastic-eating microbes as a biological way to solve this problem. There are also biological systems to potentially aggregate microplastics just so that then we can dispose of them correctly. So there are some studies looking at slime. I'm not sure about from gastropods, but certainly from these stinging animals, cnidarians. So that could be a possibility, but you'd have to ask the microbiologists working on their plastic-eating bacteria, or these waxworms, I think, are another candidate for eating plastic.
So this eating plastic is nice. It could be helped. It could be helped with this field, but is there a lot of people working on this?
I might be biased. I have a former student who's doing this now.
Okay.
There is a group actually at the Dutch Institute, the Royal Institute for Sea Research, NIOS, on the island of Texel. I know they have a big group working on this, and there's a consortium of PhD students who are all working on different aspects of this. One of my former students is doing her PhD there, and so it's fun to see as the results come in. So there are groups in the Netherlands that are working on this, but globally, plastic pollution is a big problem. There are lots of groups, from research groups at universities to NGOs, all government agencies, all sorts of different groups trying to tackle this problem. And when we think about it as ecologists looking at the ecosystem, plastics are a now very abundant, sadly, resource, and they are made out of carbon. So I think something will evolve to exploit this resource that we've dumped into
it's biomass.
Everything.
It's biomass.
Yeah. So and I think it's not yet, but it will become part of the biomass because something will learn to eat it and exploit that.
I'm thinking you mentioned different organisms, and at the beginning you also mentioned that you were working on a specific field, specific research that you were doing here in collaboration with some people in the US. So is your group very in contact with universities or research groups all over the world? Is it very interconnected? Is it very global? How does this interface work?
Yeah, so in my scientific career thus far, I've been in 6 different countries, and I definitely have connections in those countries with the groups that I worked with at the time. The collaborators from the US are a group where I did a postdoctoral project in San Diego at Scripps Institute of Oceanography. And so I did this project with them. Took years and years to work through the data. We ended up teaming up with a group from Harvard, and then it kind of all culminated in this study that we put out last year. So those are kind of, I guess the Americans that I work with, but are also other research groups throughout Europe, certainly still in Germany, where I did my PhD and some postdoctoral work as well. Still have contacts in the Netherlands, Sweden, Austria, all the other places I've worked, other labs I've been part of.
It's a pretty international field when you're looking at ecology specifically?
Yeah, I think in this particular field, but also how I designed my academic trajectory. So there are plenty of people who have international colleagues but haven't lived all over the place. I've chosen to move around a whole lot and then now settle here. But yeah, that's not the case for everybody.
What drawbacks have you found in your research career? So has been moving a lot an advantage or a disadvantage? How has this played through your career? Did it give you like a more broader perspective of how things are done? Or was it sometimes a disadvantage as in, you will come here, but then we know you're going to move quickly. So because you've been in different places, how has this played out in your...
Overall, for me, I think it's been an advantage. I like that I have an international group of collaborators that I have actually spent physical time with in their labs. So for me, that's been an advantage. But I think it could change completely depending on what field you're in. So if I was an expert on the Wadensee mudflats, well, we need those in this country, and we don't necessarily need them in other countries. That's very relevant right here. So I think it really depends what your field is. I would also argue the Netherlands is a small country, and we don't need 10 different research groups all looking at fundamental aspects of sea slug biology. I certainly don't want the competition when it comes to writing grant applications, but I don't think we need that. So I think that I have this large international group because there are 1 or 2 labs in various countries, or maybe 10 labs in the world studying things like this. And then, of course, I know and I work with all of them. But we don't necessarily need more groups like that here. But because my research, I guess, because these systems can be quite niche, it makes sense then that people are kind of scattered all over the place and not all concentrated in one spot.
This is kind of research that you can do in any lab. Like not any lab, but like it's not sea slugs from this specific region. You're looking at sea slugs from a global perspective.
Yeah, well, I would say that sea slugs are all over the place, and you don't need local knowledge to study sea slugs, because we do have representatives from almost every genus all over the place. So that means people all over the world can fall in love with sea slugs and decide to start lines of research or start labs looking at them. And so in that way, it's also great that we have an international community because we can make global comparisons or look for trends throughout the whole world.
And maybe as a final question, we've been talking a lot about sea slugs. Do you have a favorite sea slug?
Favorite sea slug? Yeah, I'm pretty partial to the blue dragon sea slug, Glaucus atlanticus. It's one of the ones that has no shell. It's able to steal stinging cells, which makes it very cool. And it feeds on...
I have the
Google the
picture.
picture. Yes, it can feed on scyphozoans like the Portuguese man-o'-war, if you know these.
Which one? These are
Like
the very long ones?
Yeah, these Portuguese man-o'-war, they kind of
Yeah.
look like a pink or blue balloon, and then they have really long tentacles.
Yes.
So they're a kind of a collection of communal organisms, colonial organisms, and people think they're jellyfish. They're not. But they float around, and they use this kind of balloon as a sail. And so that's why they call them like the man-o'-war, like the ship. And so they float around, and then these Portuguese man-o'-war sting all sorts of fish and stuff. That's how they survive. And then these weird sea slugs find them, live on them, eat them, and steal their stingers.
Oh, as well. Where can they be found?
Worldwide. Mostly more tropical areas. I don't think they're up quite this far north. But yeah, they can be found all over the place. And every once in a while, you'll see a picture of somebody holding one on their hands. And I have to just kind of, take a deep breath, because I know that they could have the stingers from a Portuguese man-o'-war, which can sting humans, give you a horrible sting. And then you'll see some, tourist or somebody finds one on the beach and holds it in their hand. And you just think, oh my, you are so lucky that you're not horribly stung right now.
So if you ever see a blue dragon sea slug,
Don't touch
Don't touch it.
it. Lots of pictures, but don't touch it.
Does it work the same way with other sea slugs? Just don't touch them.
In general, don't touch it. I would say in general with most life, unless
wildlife,
you
right?
really know the species, don't touch it.
That's a good rule of thumb, yeah.
Yeah, I think so. But certainly sea slugs, certainly when they're brightly colored, don't touch them.
They're
Don't
screaming
eat
at
them.
you. They're already screaming at you.
Right. Take lots of pictures. Post your pictures. Talk about how they look like Pokemon. All of that stuff is perfectly fine. But yeah, don't touch them. Don't eat them.
So with that, we would like to thank you, Elise. Thank you very much for being here.
Sure. Thank you for having me.
It's been a pleasure to learn about the mystery of marine biology and sea slugs, with shell or no shell. And we'd like to also thank you, listener, for being here. And we'll catch you next time. Thank you very much.
Thanks.
See you.